In this video, I want to talk about two unique bacterial structures. The first one being endospores and the second one being biofilms. We're going to cover endospores first, and an endospore is a survival structure that is formed by some types of bacteria. The process of making an endospore is called sporulation. So unlike the other bacterial cell structures that we've already discussed, like the cell wall, the flagella, the glycocalyx, etc., those structures are present all of the time in the bacterial cell. The endospore is a structure that is only made in times of need. The bacteria do not always have the spore. They make it when conditions for survival are unfavorable, and so they will make this spore, package up their DNA, save it, and hope that conditions improve enough for that spore to eventually give rise to a vegetative bacterial cell again. So we start with our vegetative bacterial cell, and that's what we call the living bacterial cell that's undergoing metabolism, is vegetative. And I've only drawn a chromosome, the cell wall, and the cell membrane for this bacterial cell. So we're going to just imagine that the cell is living, its conditions have gotten really harsh, and it's facing either sporulation or death. Now obviously it wants to continue to survive, so it's going to undergo a sporulation event. The first thing that happens is that the cell will make a fresh copy of its DNA or its chromosome. So it makes new DNA for this. And once that new DNA is made, now this cell membrane is sort of going to pinch inward, and we're going to have a cell membrane surrounding this new DNA. We're going to have cell membrane around the old DNA. So we have cell membrane around both. And the next thing that is going to happen now is we don't need this old DNA anymore. And so it's going to be chopped up into little bitty pieces. It's going to disintegrate and go away. So now I have a situation where I have this fresh DNA inside a cell wall and surrounded by two membranes, because the new DNA is also going to kind of steal the membrane from around the old DNA. So I have DNA surrounded by an inner membrane and an outer membrane. And so now these are going to be called the inner four-spore membrane. Some really bad handwriting. And the outer four-spore membrane. Now these endospores will persist in the environment for decades, sometimes hundreds of years. Some scientists think they might survive millions of years or more and still be viable. In order to be able to do that, they have to be very tough. So directly to the outside of this inner four-spore membrane now, the cell is going to deposit a layer of peptidoglycan. So we've already learned that peptidoglycan is pretty tough, and it can withstand a lot of osmotic pressure. It's a very strong substance. So we get this layer of peptidoglycan deposited here outside that membrane. And then to the outside of the peptidoglycan, between the peptidoglycan and the outer four-spore membrane, we're going to deposit another very tough substance, and that is protein. So we know protein's pretty tough. And so each of these substances are resistant to different types of environmental forces. And so this gives the cell a lot of resistance to things like drying out, to things like UV light, heat, chemicals, and so on. And so if I kind of draw this guy again with our chromosome, our inner four-spore membrane, our layer of peptidoglycan, and our layer of protein. Now the outer four-spore membrane is going to accumulate a lot of wax. So the outer four-spore membrane now becomes the spore coat. And it's very, very waxy. And this again contributes to the ability of the cell to resist desiccation. And so we've got our DNA surrounded by a membrane, surrounded by peptidoglycan, surrounded by protein, and ultimately encased in wax. So think about cheese. Sometimes cheese is preserved by being dipped in wax. It acts as a preservative. It keeps things out. It keeps the moisture in. So this works really well to preserve the chromosome inside of these other substances. And then once that outer four-spore membrane has been converted into the spore coat, the cell wall of our bacterial cell disintegrates, and we're left with the endospore. And as I mentioned, these endospores can persist for a very long time, for decades, sometimes hundreds of years in the environment, and they are resistant to a number of things. So they're resistant to nutrient deprivation, so there's no food, so what? We'll just make an endospore. They're resistant to desiccation, which is drying out, so there's not enough water. They're resistant to chemicals like disinfectants, antiseptics. They're resistant to UV light, and they're resistant to heat. So endospores are very, very difficult to kill. So they are notable in food preservation situations because they are resistant to heat. The spores, if the food is not cooked correctly, the spores will germinate inside the food. Often they contaminate canned food, and this is where we get diseases like botulism. So botulism comes from the bacterium Clostridium botulinum, which frequently contaminates canned goods. If this bacterium isn't killed during the canning process, it germinates inside the can, it grows, and it produces botulinum toxin, which causes flaccid paralysis. And if enough of this is ingested, it paralyzes the diaphragm, which prevents the victim from breathing, and then they suffocate to death. Unique use for botulinum toxin, you might recognize this, Botox, injected in people's faces and other muscle groups cosmetically to prevent the facial expressions that cause wrinkles, but it can also be used therapeutically for people who have uncontrolled muscle tics to give them some relief from that. It paralyzes those muscles so that they're not spasming all of the time. So once the conditions become favorable again for the cell will germinate and become a vegetative cell again. The process by which it does this is not as well understood by scientists as the process for sporulation. It's very difficult to study because these endospores do not produce any discernible metabolism. So we have a pretty good understanding of how the spore forms, not as good an idea as how it goes from being a spore to a live bacterial cell, just that it can do that once the conditions become favorable. Finally, the two most notable genera that form endospores, I already mentioned Clostridium, and the other one is the genus Bacillus. And this one is notable because of the microbe Bacillus anthracis, which you may recognize as the bacterium that causes anthrax. Anthrax can happen naturally. It is a disease primarily of livestock, but Bacillus anthracis is used for bioterrorism because the endospores have a consistency similar to talcum powder, and disseminating the spores into ventilation systems, into water supplies, is really quite easy for bioterrorists to do. People who contract Bacillus anthracis need to seek medical treatment immediately once symptoms appear. It's too late. The disease is fatal. I guess the silver lining to the anthrax cloud is that the disease cannot be transmitted from person to person. The only way to contract anthrax is to be exposed to the bacterial spores directly. Our next structure is something called a biofilm. And a biofilm is defined as a complex aggregate of microbes living together and surrounded by a polysaccharide matrix. Now, it is important to note that this polysaccharide matrix is the same stuff that the glycocalyx is made out of. It's the same polysaccharide. But instead of surrounding only a single cell, the matrix surrounds the community. So how does the biofilm form? We have a solid surface. This could be a microscope slide. It could be your teeth. It could be your shower wall, a rock in a stream. In the medical environment, it could be inside a urinary catheter or any type of indwelling medical device, any type of implant catheter, or something like that. Any solid surface these microbes can attach to. So the microbes attach to the surface initially by weak chemical interactions called van der Waals forces. And once they are weakly attached, they are going to send out chemical signals to attract other microbes. And so now this is going to bring in, okay, I had to pause to yell at barking dogs, but continuing on, these signals for attraction are going to bring in other types of microbes. Now these could be bacteria, or maybe we get some algae, which are quite large. We might get some prokaryotes. So we get some different types of microbes being attracted to this community, and they come in and they begin to attach. And then the bacteria begin to secrete the polysaccharide, which then surrounds the entire community and protects this community. So just to sum up, the microbes attach to the surface. They attract others, and then they secrete the matrix. Now, the benefits of living in a community like this is the matrix is protective. So once inside this protective matrix, waste products from one microbe might serve as food for another microbe. They can share genetic information, and they are protected from chemicals. They're protected from being removed from the surface. They're very difficult to remove. Antibiotics can't get through the matrix to them. Disinfectants, antiseptics can't get through that matrix. So they have to be removed mechanically. So even though you can't see them, and you might spray them with Lysol, you might spray them with bleach, you have to scrub them mechanically to remove them. And because their removal is so difficult, that's why they're a huge problem in healthcare, because they get into primarily urinary catheters. They're a huge problem in those because there's a lot of nutrients in urine. They form on the catheter, very difficult to get rid of, and they cause infections, kidney infections, urinary tract infections, but they can form on other indwelling devices as well, such as pins, artificial hips. If someone were to undergo heart surgery, they can form on artificial heart valves and so on and so forth, and cause a number of opportunistic infections in people who are already immune compromised. So they are significant in that they are very difficult to get rid of.